All right, welcome. Today we are diving deep into a mystery from the 19th century. One that, once we crack it, reveals the simple, elegant rules that literally write the code of life. So, let's kick things off with a question that, applies to every single one of us. You've got your mom's eyes, maybe your dad's nose, but you're not a perfect copy, right? You're a unique mix. So where does all that variation actually come from?
Well, the answer, believe it or not, starts in a garden. And not in some high-tech lab, but in a monastery garden with a monk named Gregor Mendel. Yeah, between 1856 and 1863, this guy meticulously crossbred thousands and thousands of pea plants, all to figure out the basic rules of heredity.
Okay, so let's dig into the first piece of this puzzle. The thing that made Mendel so brilliant was his focus. He didn't try to look at everything at once. He chose to look at simple, really clear-cut opposing traits.
So he took what we call true breeding plants. What's that mean? Well, a tall plant that only ever makes other tall plants, and a dwarf plant that only ever makes other dwarf plants. He crossed them. And get this, in that first generation of offspring, we call it the F1 generation, every single plant was tall. Not kind of tall, not a mix, all of them tall.
And that right there was the big mystery. It's like the dwarf trait just poof, vanished into thin air. It wasn't blended in to make medium-sized plants. It was just gone. So what in the world happened to it?
But, and this is where it gets really interesting, Mendel then took those tall F1 plants and crossed them with each other. And in the next generation, the F2, guess what? The dwarf trait popped right back up. It reappeared totally unchanged. And what's more, it showed up in this really predictable pattern. For about every 3 tall plants, there was 1 dwarf plant.
So, to explain this, Mendel came up with an absolutely brilliant idea. He figured that inheritance isn't like mixing 2 cans of paint. It's not about blending. Instead, it's about passing down these invisible, separate little units. He called them factors. Today, of course, we call them genes. These are the basic units of inheritance, the little packets of information that carry instructions for a specific trait, like, plant height.
And these genes, or factors, they can come in different flavors. We call these different versions alleles. So for the gene for height, there was a tall allele and a dwarf allele.
And this leads us to a super important distinction. You've got the genotype, that's the actual genetic code an organism has, like the letters TT or T small t. And then you have the phenotype. That's the physical trait you can actually see, like the plant being tall or dwarf.
And this is the key that unlocks that whole first mystery. Mendel realized that one allele can totally hide the effect of another one. The one that shows up, he called dominant, and the one that gets hidden, he called that recessive. So a plant that has 1 tall allele and 1 dwarf allele is going to be tall, because the tall allele is dominant. The dwarf trait is still there in its genes, you just can't see it. Okay, so the dwarf trait was hidden, but it wasn't gone. So how did it pop back up in that second generation? To answer that, Mendel had to figure out the fundamental rule of the game, what we now call his first law of genetics.
He called it the law of segregation, and honestly, it's beautifully simple. Here's the deal. Even though a plant has 2 alleles for a trait, those 2 alleles get split up. They segregate when it creates its reproductive cells. So each little sperm or egg cell, what we call a gamete, only gets 1 of the 2 alleles. There's no blending, no mixing, just a clean 50-50 split.
This law is the absolute key that unlocks the whole puzzle. And luckily for us, there's a simple tool called a Punnett square that lets us see exactly how it all works. Okay, so let's go back to those F1 plants, remember? They were all tall, but they inherited a tall allele from one parent and a dwarf allele from the other. That means their genotype was heterozygous, 1 big T, 1 little t. Now, according to that law of segregation, when these plants make their gametes, half of them are going to get the big T allele and the other half are going to get the little t allele. Makes sense, right?
So, with the Punnett square, we can map out every possible outcome. We just put the possible gametes from one parent along the top, a T, and the gametes from the other parent down the side. Now, we just fill in the squares. The big T gamete can meet another big T to make a TT offspring. A big T can meet a little t to make TT. And finally, a little t can meet another little t to make TT. And look at that. This explains exactly what Mendel saw in his garden. If we look at the physical traits, the phenotypes, 3 out of the 4 squares give us a tall plant.
the TT1 and the two TT1s. Only one square, the TT, produces a dwarf plant. And there it is, that classic 3-to-1 ratio. The mystery of the reappearing dwarf trait? Solved.
But wait, let's look a little closer at the genetics, at the genotypes. This is the really crucial part. We have one TT, which is homozygous dominant. We have two TTs, which are heterozygous, and we have one TT, homozygous recessive. That gives us a 1-to-2-to-1 ratio. That's the underlying genetic truth that creates the 3-to-1 result we see on the outside.
So why are we talking about a 150-year-old experiment with pea plants? Why does this still matter so much? Well, because these principles he discovered, they're basically universal.
Mendel's ideas gave us some incredibly powerful tools, like something called the test cross. Imagine you have a tall plant. Just by looking at it, you don't know if its genotype is TT or TT. But if you cross it with a dwarf plant, which you know has to be TT, the babies will tell you the secret. If any of the offspring are dwarf, well then you know the tall parent must have been carrying that hidden recessive T allele. It's kind of like a genetic paternity test for plants.
The crazy thing is, Mendel couldn't see genes. He didn't know about DNA. He was just working with patterns he could observe. But his revolutionary idea that inheritance comes in these little packets, these discrete units that don't blend, that idea completely set the course for the next century of biology. It's the very question that eventually led scientists like Watson and Crick to discover the double helix structure of DNA itself.
So Mendel's work handed us the key to understanding heredity, everything from modern medicine and curing genetic diseases to engineering the crops we eat. We are still exploring all the doors that were unlocked by a monk who just wondered why some of his pea plants were tall and others were dwarf. The only question left is, what are we going to do with that key next?